Hi, this is Talk on NAVUKA, a channel where we'll talk about science with researchers in simple terms. My name's Yanina, and our conversation today is with Dr. Haydn Kissick about innovative research in cancer treatments. The video has subtitles in Belarusian, English, and Russian. Click below to subscribe and leave your feedback in the comments. Hi, Dr. Kissick. Thank you for joining us today. And do you mind telling us a little bit more about your journey into the science, how it began, and what did you do?
Sure. So I was a PhD student in Australia. I started my PhD in around 2005, I think, maybe 2006. And around that time, I'd taken some immunology classes as an undergrad, and I just thought it was very interesting. And I spoke to a lab. They had a PhD position open, and cancer immunology was the topic that they were working on, and that's what I got drawn into. I studied how a cell called a regulatory T cell stops the immune system killing cancer. And then around 2010, immunology to treat cancer became a new way to treat this cancer. There was a very important drug approved called anti-CTLA-4 that was really the start of a sort of recent revolution in cancer treatment. And this was all happening in the United States. So I decided, with my wife, who was also a scientist, that we really wanted to move to where most of the science was happening. And then around 2014, I came to Emory. Emory is very well known for their immunology program. It's a very big center for immunology research. It's right next to the CDC. They have the Vaccine Center, which does a lot of studies on vaccines to infectious disease, not so much cancer. And I was one of the first cancer immunologists that came to Emory. And in the last 8 years since I've been here, many cancer immunologists have joined. And now we have a really big program studying the immune response to cancer, how we can treat patients. In the 10 years since 2010, when I told you that this first drug was approved, it's now the main drug used for most cancers when patients have advanced disease that is life-threatening. We use a patient's immune system to cure the cancer in many cases. And the next 10 years learning how we can do that better is really what I'm focused on. It's what I started on. I'm still interested in it. And I really think it's a new way to treat cancer that is becoming an impressive treatment for patients. And it's not as bad a disease as it was 10 years ago.
What is the immune system, if you can just talk about it briefly?
Sure, I can talk about it briefly or in a very long way. Your immune system's job is to kill anything that enters your body that's really not meant to be there. And it's constantly surveilling and looking for things that aren't meant to be there. So when you cut your finger or something, almost always some sort of bacteria or pathogen makes its way into your blood, and the immune system has to be ready to kill immediately. So in the blood, there are cells that are part of your immune system just constantly circulating and constantly testing any area to say, is there anything here that is not my own tissue? It's very good at sensing its own tissue and something that's not its own tissue. And as soon as it finds something that's not it, we call it self, not your own cells, it activates extremely quickly. A few minutes, the cells will start dividing, there'll be more and more cells, and then they'll kill anything that is not meant to be there over the course of a couple of days. So that's for a bacteria, things like that. Cancer's a little bit different because it's your own cells. So you're not sick with a virus or a bacteria or something like that. Your own cells have started to do the wrong thing, usually grow too much. And so that's a bit of a challenge for the immune system because it's meant to not kill your own tissue. If it does that, you die. So for many years, probably more than 100 years, people didn't believe that the immune system would kill cancer. But around the 90s, late 80s and 90s, we started to see that, well, really your immune system, when we take tumors out of patients, there are a lot of immune cells as part of the cancer. They're obviously responding in some way. And then, through the 90s and then early 2000s, they really started working out that, yes, like even though it's your own tissue, the immune system does find a way to actually kill those. And basically what's happening is through your life, you're continuously getting small bits of cancer and the immune system kills it and you never even know about it. So it's kind of taking care of this cancer as all the time. And that's what we're sort of thinking about with immune response to cancer. How do we make the immune system sort of see that cancer as something that shouldn't be there and then kill it and get rid of it? It's a very fine balance that we're sort of, in all the immune diseases, that we're sort of constantly trying to turn on and turn off so that we can help autoimmunity, help transplantation, or turn the immune system on to better kill cancer, better attack viruses, and do things like that.
You mentioned that it's able to evade the immune response.
Yeah.
So does it mimic that it's more self than it was? Or what
That's a really great question. So I'll talk about, there's lots and lots of examples, but I'll use the one that we use as one of our therapies now. So there's this molecule, it's called PD-L1. It's a programmed death ligand 1. It was, discovery of it was a Nobel Prize a few years ago. And what this usually does is in your body is it's on tissues that might have some immune activity near them, but you don't want your immune system to kill. So things like the gut has a lot of bacteria in it. And your immune system has to not attack all those bacteria because they're actually good, they're helping you, but they're still foreign. So one of the ways the body deals with this is it has these proteins on the surface of cells that just, when immune cells get there, it says, okay, don't kill me. Like, I know you're all active and you've seen something, but here is not the place to be killing. PD-L1 is one of these molecules that does that. But that goes wrong in cancer, where the cancer cell hears about that trick and it thinks, okay, well, that would be excellent. If the immune system comes here and I have PD-L1 on me, it'll get tricked into thinking, okay, nothing to see here, I will not attack. And that was a very big discovery because now, tens of drugs from almost every cancer, if we just block that molecule on the surface of cancer cells, now the immune system gets to the cancer and that signal that's telling it to stop is blocked and the immune system just kills the cancer as if it has nothing there. So these mechanisms that normally protect us from autoimmunity, the cancer sort of takes those and uses them to protect itself from the immune system.
And is every cancer has this PD-L1?
Yeah, no. So not everyone. Like I said, there's many ways a cancer can trick the immune system. For our patients in most cancers, around 20% of the patients, if we give this drug, they have an incredible response. Immune system clears the cancer, takes out metastases, all sorts of things. But for a big proportion of patients, it has a small effect or sometimes no effect at all. So those cancers are probably using a different trick. And PD-L1 is not something that they decided to use, and then the drug doesn't work. So we have to kind of work out all those different things and how we can, outsmart the cancer, I think.
What does make this drug an immune therapy? Is it what is it? Is it similar to chemotherapies in any way, or it's somehow entirely different chemistry?
Chemotherapy drugs are what you might think of as like just a chemical. And they typically enter the cancer cell and they're just very toxic to the normal process of a cell. And so the cancer cell starts dying because this molecule is in it and it can't, function normally. I think around the late 90s, we started making drugs that are something called antibody drugs. And these are very different. So they're a part of the immune system, so it's a bit confusing. The immune system usually makes antibodies, but we can take those antibodies and use them for useful things. So I think the first human one was rituximab, which is an antibody that binds to a type of cancer cell, and then just it kills the cancer cell. And an antibody is a very useful tool. It is very specific for, a certain part of a cell or a certain part of a bacteria. It will bind only to that, doesn't go everywhere. So if you think about those cancer drugs, they're going into every cell. You're getting very sick because, your liver cells are dying, your kidney cells are dying. But a drug like this an antibody only goes to the thing that you pick. And so this PD-L1 drug, it only sticks to the PD-L1 molecule. It blocks it. It just sort of sticks itself onto it. Other things can't interact with it. And that allows it to be less toxic than the other drugs. It's not non-toxic. Okay, so your immune system is very powerful. It can kill you in a couple of hours. When people die of influenza, it's because the immune system is doing too much killing. And in the case of cancer, this PD-L1 is doing important things all over the body. The gut's protecting the gut. It seems to have a very important role in the heart. And so when our patients get put on this drug, sometimes they have very bad side effects. Their gut gets very inflamed because the normal role of this molecule is to protect your own tissue from your own immune cells. We've taken that away. Now the immune system attacks the gut. That's not good for patients. Over the long term, we're sort of starting to see that some of these patients have small heart problems where the immune system starts to cause some problems with the heart. And we don't know why. We need to understand much better why that happens so we can, even though it's very targeted, we can sort of at least understand and maybe try to overcome those problems.
I read that some antibodies are conjugated to the chemotherapy molecules. And is that beneficial, or is just for the cases when it's really hard to treat? Yeah, in fact,
so going back to the chemotherapy, how it works, usually it's just a drug that goes in the whole body and hopefully it finds its way to the cancer. And that's very toxic for the patient. But if you take these antibodies that will stick to a very specific thing, you can make them against the cancer. So you can make an antibody that might bind to something on the cancer that is really specific to the cancer. You can tie the drug to the antibody, and then it'll just go straight to the cancer and hopefully only go to the cancer cell. And there's been a few drugs approved for that recently. There's a lot of challenges. It's kind of hard to get the antibody to go into the cancer and not just everywhere else. But it's starting to work. It's a good approach, and some interesting things there. It reduces the toxicity of the toxic drug to the other cells, basically.
What about the T cell therapies?
Is
that immune?
I forgot about a whole important arm of immunology. This is a class of drugs called cellular therapy. So all the things I've kind of talked about up to now are things that are, we give you a drug and it affects your immune system, and then the immune system kind of kills the cancer. A different approach, it's been going on just as long as a lot of these things, is to take a patient's immune cells from their blood or their cancer or something like that, and then take them to the lab and then just expand them a lot. So they'll take, sometimes a few thousand cells, maybe 10,000 cells, and they'll expand them over the course of a few weeks to billions and billions of immune cells, and then inject them back into the patient with the idea that these are the cells that kill cancer. If we have a lot of them, it'll work. And in some cancers, it's just incredible. So this was all done at the NIH. For their melanoma patients, around half the patients, it completely cures. The challenge is getting the cells from a patient's tumor, growing enough cells for that patient, getting them back into the patient. It's a very labor-intensive and difficult process to do. So based on that work, people tried to then take a few immune cells from any patient and genetically engineer them to be kind of like the good killing cells, and then use those cells to kill the cancer. And that approach has worked pretty well for a lot of the blood cancers. I forget exactly which ones it's successful and which ones it's not. But this is one of the new treatments now for blood cancers. They take your own immune cells, expand them, genetically engineer them, put them back into the patient, and then they kill them. Hasn't quite worked for many of the solid tumors yet. But the hope is that the same approach can be used, and that's a massive area of research now.
Can you please talk a little bit more about what you do in your research and how is it translated into clinical applications?
Right. So we're very basic research lab. We don't necessarily have the goal of turning what we do into treatments. We're trying to just understand better how the immune system fights cancer. Inevitably, it does turn into treatments and things like that, but we sort of start with these very basic questions. So right now we're looking at the cancer of patients who have a very strong immune response. So some patients, you take their cancer out of them, they have surgery, remove the tumor, you look at the immune system of these patients and it's just incredibly strong. You know, there's all sorts of immune cells in the cancer, you know, doing a good job trying to control the disease. And these patients across many cancer types do very well after surgery and other treatments. The immune system is helping, basically. Then there's another group of patients, and it's sort of 50-50, who have almost no immune response at all. There's no immune cells in their tumor. It's almost like it's completely unobserved by the immune system. So, and those patients do much worse typically. After the surgery, sometimes metastatic disease will pop up. If they go on therapy, they don't respond as well. And so we have a very simple question, which is, why does some cancer have this amazing immune response and the patient do well, and some patients have no immune response at all? And we study that in many ways. So we've studied the genetics of patients' cancers. Maybe this particular cancer is different genetically than the ones that have an immune response. Yes, there are some really interesting genetic changes that make a cancer hot or cold, which is a big part of our lab's research now. What exactly do these genetic changes mean? Why do they trigger an immune response or not? And then so that's from the sort of cancer side. What has the cancer cell done? And then we study the immune side as well. What exactly do immune cells have to do to kill a cancer cell? It's not as simple as just, you know, turning up and killing. There's many steps involved that the immune system has to kind of go through to actually kill a cancer cell. So we're really interested in just understanding those steps, right from the very early time there was first a cancer cell to, you know, years later when there's this very powerful immune response. What were all the steps that had to happen so that you could get this really big immune response to the cancer? And that's a little bit more where the therapy comes in, right? Because if we find one of those steps is broken, then we can start to think about treatments to kind of fix that particular step. We have one recently that we think is important. The T cells, when T cells are the main cell that kills a cancer cell of your immune system, when they get to the cancer, there needs to be something like a danger signal that tells the immune system to kill. It doesn't kill unless there's some signal that says there is danger here. Kind of like when a virus infects you or a bacteria infects you. And it seems that some of these patients don't have any of this danger signal in their tumor. And so we're doing a clinical trial now where we sort of inject the tumor with basically parts of a virus to make the cancer seem more dangerous to the immune system, and then it activates. It's kind of funny because it's one of the first immunotherapies that was ever tried hundreds of years ago, more than 100 years ago now. One of the first immunotherapies was a doctor injected a tumor with boiled bacteria, and the tumor was cured. And that was basically the start of our immunotherapy idea. Now we're doing it in much more sophisticated and detailed ways, but it's kind of the same idea. So basically what we learned is taking us back 100 years to do the same thing, just with some more knowledge and a little bit more refined approaches and things like that.
So do you inject that in patients?
Yeah. Well, the clinical trial, we tested it, so I can tell you the process. We had the idea. We studied many of these steps involved in immune response to cancer. We sort of worked out a place where it seemed to be going wrong. We tested this approach in animal models, mice, things like that, to show that it sort of worked. And then there was a drug company who were already making this drug and trying it for slightly different things. And then we said, we think this will work pretty well for cancer. So now in a small group of patients with prostate cancer, metastatic prostate cancer, probably later this year, we'll enroll the first patient, and we'll be seeing if this sort of approach, fixing this problem that we think happens right in the cancer, will help us boost the immune response to the tumor. We hope that it is the fix, but realistically, it's going to be many steps of us trying these treatments, doing our very best, and then learning how the immune response changes. Did we fix it? How did we fix it? Did the drug do what it was meant to do? There's many steps in kind of getting all this right before it's like a good treatment for patients. And
So just to make sure I understand correctly, you inject the drug into the existing tumor?
Yeah.
Okay.
Yeah. And so these are patients who have a tumor in a region where it's very difficult to maybe surgically remove it. But it's quite, I'm not going to say quite easy, it's relatively easy to take a small needle and get it to the tumor. And so it goes straight to the tumor, it's injected into the cancer, and then hopefully activates the immune cells that are already there to activate and do the right thing to kill the cancer cells.
And so the patients whose immune system is not as responsive to cancer probably not going to benefit from this, because
we hope this is one of the, we actually hope that this is, we're focusing on the patients with a bad immune response actually.
Oh, okay.
And because we hope that this is one of the things that is the reason why they have a bad immune response, and then we can kind of fix that particular type of cancer, fix these ones where the immune response is not doing a good job. The patients with a good immune response don't need as much help. A lot of them survive, indefinitely after their surgery. Or if their cancer does come back, our current drugs are actually really, really good at using the immune response they have. It's really the patients whose tumor for some reason doesn't have a good immune response. We need to make drugs for those so that we can fix the problems that went wrong and get them into the same sort of responding to these immunotherapies.
And do you work with cancer in cell lines, or do you use animal models primarily?
Lots of different ways. Everything starts with the patient's tumors. So we start by looking at a human's disease. If you start with the animals, you might get a funny answer, and then it's not real in patients. So we get patients volunteer to give their tumors after surgery to us for research, and then we can study lots of features of their immune system to kind of learn about what happens. So that's the first place. We want to see, what does an immune response look like in human cancer? Then animal models come into it in very important ways, because we have to try a lot of things. We have a lot of ideas. We get that data from the patients, and maybe 5 or 10 things occur to us that is maybe happening. But we can't go and test those in patients because they may be dangerous. There's a good chance that our ideas are wrong and they don't work, and that's often what happens. So the animal models allow us to try many things and work out which of our ideas might be correct. So we do that. And then we've kind of got this new system which is in the middle, where we actually take patients' tumors and their immune system and we can grow them in animals. So now a patient's, their own cancer, we take a little piece, we put it into a mouse, it starts growing in the mouse. We take the patient's immune cells, we put that into the mouse, and we can almost study how that particular patient's cancer and immune system is going to respond to different ideas, different therapies. And if we change the system, how does the immune response change using patient's tissue? It's sort of a hybrid system that gets in the middle. It's a bit closer to the realistic human disease. We can do a little bit less with it though. So that's sort of how it works for our research. As I said, we don't start thinking about, let's make a treatment for cancer. We start thinking, let's learn about cancer. Let's learn about how the immune system responds to cancer. And so in doing that, we learned, in the last 5 or so years, our lab of just studying the immune response to cancer has learned a lot of things that we didn't know. And then our next thought is, like, once we learn something new about the immune response to cancer, we think, well, is this something that we could change to try and treat a patient? So we start with the base level of knowledge, like we learned something new about it. Can we use this to treat cancer? Sometimes and often the answer is no. We learned something and it's really interesting and we don't quite know how we would use that information now. Maybe in 10 years it's useful, maybe it's never useful, we don't know. For this particular one, we sort of found this thing in the tumor that seemed to be a major limit to the immune response to cancer. So we think that that's a pretty good bet for something that we could try and fix and hopefully make a better immune response to the tumor.
And is the cancer treatments personalized, or do they tend to become more personalized than they were?
Yeah. So, the idea of personalizing a treatment to a patient is hopefully you could pick the best treatment that cancer and patient is going to respond to, so that we don't have to try all the drugs. The typical way in the past for treating a patient was, they'd come in, we'd try the best drug first. Some patients respond. After 6 months, we work out that a patient doesn't respond. We try the next treatment. Some respond, some don't. We try the next treatment. And for patients, this is a terrible experience. They go through many rounds of treatment. It's not working. It's very frustrating and upsetting that you're going through these sometimes very toxic drugs for no benefit. And the idea of personalizing is, before we even start, we could pick from that big toolbox of drugs which one you should get that's going to give you the best chance to respond to therapy. So there's a lot now in us trying to learn about which treatment a patient should get. And so for these immunotherapies, we sort of know if you have a good immune response to your cancer, PD-1 blockade, those therapies will work pretty well. And we sort of know if you have a really bad immune response to your cancer, they work less well. So the personalized aspect of this is, we think that if you're in that group of patients that have a worse immune response, this therapy we're giving will maybe try and fix it in a different way. So it's personalized in that sense. At the start of any drug development, we're typically working with patients who have tried all the other therapies, and they're now on a clinical trial, to test if maybe this experimental therapy will work for them. Sometimes it does, and sometimes it doesn't. So it's very much a learning process when you're in these clinical trials. Which patients maybe it does work for, so we can pick the right patient in the future, give them the best chance to respond to something.
Could you please summarize the existing treatments and what
Sure.
may be the next?
Sure. So, immunotherapy has been around for a very long time. We've been trying this for well over 100 years. Maybe since the mid-'80s, we had a little bit of success. And, the treatments that have broadly come since then sort of fall into a few different ranges. There's treatments that activate your immune system, try and boost the strength of it. So there are things like vaccines, which, we take part of the cancer, we boost your immune system against that. There are things called cytokines, which, tell your immune cells to activate and proliferate and, make more cells. Those have worked a little bit. We got some of those drugs to work well, and, so we use some of them well. The vaccines, we haven't had a lot of success with. There's a class of drugs we call checkpoint inhibitors. And so rather than activating the immune system, we're taking away the sort of suppressor of the immune system. Your immune system is suppressed in many ways to stop it attacking your own tissue. If we block the suppressors, then hopefully we get better killing with the immune system you've got. And they're very successful across many different cancer types. That's been our big win in the last 10 years, and many cancers are treated like that now. And then a little bit, newer in terms of approval, but, we've been trying it for maybe longer, is what we call cellular therapies, where we take a patient's immune cells from their body, we try and either expand them, genetically engineer them to make them really good at killing cancer, and then take them back, treat the patient with their own cells. And I do think cellular therapy is maybe, it's had some really big wins in some cancers, and it's not so successful in other. But I do think it's a very versatile treatment. Genetically modifying a cell to do the thing you want is a very powerful tool, and as we learn to use that tool in better ways, I think we'll come up with some really interesting ideas to use a patient's own cells to kill the cancer, out-trick it. It comes up with a lot of tricks to escape the immune system. And if we can kind of take the immune cells out, give them some new tools to go and fight it, send them back to the war, I think we can come up with some really great ideas to do that and kill the cancer. As for the next generation, it's hard to guess. So, like I said, we do research with questions in mind, kind of hoping that they'll be useful in the future, but we can't guess what's going to be an important finding. The discovery of these molecules, the checkpoint molecules, was not at the time groundbreaking research. And it was actually quite ridiculed. People didn't really believe it very much. Everything was about how do we activate the immune system, and we didn't really know about these negative regulators. And the scientists who did this, people were like, yeah, it seems stupid, it doesn't seem like a good idea. So, and then 10 years later, it starts getting into patients and it's having amazing effects, and all of a sudden it's not so stupid anymore. So it's really hard for us to guess what the next thing is going to be. It's why basic research is so important. Just learning about cancer and how it operates will potentially lead to things that we can't even imagine. And there'll probably be something, immunotherapy is our biggest thing right now. For the last 10 years, it's been the biggest thing. The next thing might be completely unrelated to the immune system. Could be something we don't even know about yet. And, someone maybe has discovered it already and is being ridiculed for what they say. And, but over time, if the idea is strong, hopefully we can turn it into new ideas that we use to treat cancer.
And as we were talking about immune therapy and all those treatment for cancers, the brain tumors, do they respond well to those therapies?
Well, yeah, yes. Surprisingly, yes. We didn't think that the brain would. The brain is something that people called immunoprivileged, like the immune system doesn't go to the brain. You don't want immune cells in your brain. And, in the last, 10 plus years, it's just 100% wrong. There's almost like a brain-specific immune system, which is, a ton of research is going on into that in the last 10 years. Very exciting stuff. One of the most responsive tumors to these PD-1 therapies is to the responsive cancers to the checkpoint therapies are some of the brain cancers. So melanoma goes to the brain very often. And the patients who get these checkpoint therapies, many of them are cured. There's some combination. Sometimes they need radiation to the brain and this. But they're sometimes cured by these drugs. And 10 years ago, that was you were given 3 months to live. And now a good proportion of those patients actually do very well. So very exciting sort of times for these drugs and things like that. And we don't quite know why. We don't know why immune cells go to the brain differently. Our lab is doing a study on this right now. We've looked at hundreds of patients with brain metastases, and the immunology that we see in regular tumors, kidney tumors, lung tumors, seems to be exactly the same in the brain. The immune system maybe doesn't know that it's in the brain. Maybe it just says there's cancer, I will do what I'm meant to do, and takes care of it from there. So yeah, very interesting and exciting times.
With chemotherapy, patients have to undergo several rounds of treatments, and often if the first line of treatment is not successful, they have to go for the second, third one, and so forth. Is it the same way for immunotherapy?
Yeah, it's almost more difficult to know when to stop. So with chemotherapies, unfortunately, we stop when the tumor is gone or when the patient is too sick to continue receiving the drug, which is a horrible way to give any drug. Because the immunotherapies have less side effects, we don't actually know when to stop. And it's a big question right now of when do we take a patient off immunotherapy. So if we give them these drugs, sometimes it's every 6 weeks, a month or so like that, the tumor's gone away, but there's no side effect from the drug. Why not just leave them on it. And doctors are hesitant to take you off a drug when your cancer has disappeared and you're having no side effects, everything's working well. So it's a difficult problem. And now we're starting to see that some of these patients who are on the drug for a long time are starting to develop some side effects. I mentioned there were some heart problems and things like that. So now we're sort of realizing, well, maybe there is long-term side effects. It's definitely better than cancer, but we don't want to make anyone sick. So it's a hard question to know when to take people off a drug with minimal side effects that has controlled their cancer. And there's lots of trials. Doctors are trying to do these things like, should we take you off after 1 year? Should we take you off after 2 years? And it's challenging, and there's a lot to learn there when to stop.
And would patients get the immunotherapy after having the chemotherapy previously? Often that's the thing to do. And very interestingly, sometimes the two together are much better than either one alone. So sometimes a patient maybe had chemotherapy as their first treatment, maybe nothing happened, it did nothing to the cancer, and so the doctor decides that immunotherapy is the next best option. So they get that, and sometimes they respond amazingly. Sometimes, again, it doesn't work. And now, with cancer, we really want to throw everything we can at it. We don't want to sort of play it cautiously. We want to give the best possible approach, take it out as quickly as we can. And so now people are trying things like give chemotherapy, give immunotherapy at the same time. And for some things, it's amazing. There was a thinking that it wouldn't work well because chemotherapy typically hurts your immune cells. It works by killing cells that are dividing quickly. Cancer cells are dividing quickly, but much faster than the cancer cells are the immune cells. They divide extremely quickly. So the chemotherapy often hurts the immune system. So the thinking was these two won't go well together. But they kind of do. So we have a lot to learn there. We don't know why. Maybe it brings it down a little bit, but the drugs that activate it activate better. It's a big area of study. How do we put the tools that we have as cancer drugs, how do we put them together in the best way to kind of cure the cancer? I'd say that's probably the biggest area of research right now. We kind of know these checkpoint therapies are good, but how do they work with all the therapies we made in the 50 years before that? Which combinations of drugs might work well together to really sort of synergize and kill the cancer better? And due to the COVID, now everyone knows that the viruses mutate and they can escape the immune system, and the same goes with HIV patients. Can cancer do that too? Yeah,
I'd say that is the feature of cancer. Cancer's the whole disease is mutating and evolving. So the start of getting cancer is your cells have to divide to maintain your body. You've got a liver cell or a lung cell or something, one of the cells died and the cells near it need to divide and replace the ones that died. And it's a big job. So you've got to copy all the DNA. There's 3.5 billion little molecules that encode your entire instructions for your cell. It has to make a copy and then pass it on to the cell that it gives rise to. And if it makes a mistake, then all of the cells that come from that new cell have the mistake copied over. And there's a lot of checking and making sure everything's okay. If these systems that check break, now mutations start entering into the cell and they start messing things up. So that's usually bad for a cell. Usually you break something important and that's terrible, or the cell dies. But sometimes you break something that was like a negative regulator of division. There's lots of things that say, don't divide. Don't need to divide now, everything's fine. If you break one of those things, now the cell's like, it's fine, I'll keep dividing. And it'll just keep dividing. That's still not cancer. That's a dividing cell. Everyone kind of has that sometimes. But the mutations are creeping in, and it's an evolutionary process where there's selection for cells that have an advantage over the cells around them. So now if a cell becomes good at recruiting blood vessels, it can survive better because it's got blood coming to it and it can divide more. It might recruit, mechanisms to get more sugar. So now it has more energy and it can do more things. Then it eventually uses up all the resources in an area. It might gain the ability to go into the blood and move somewhere else. And so there's this constant selective pressure to select for variants, tumor cells that are better at surviving in the host. And one of the things that gets selected for is the cell's ability to not be killed by the immune system. The immune system's constantly killing. It's like if there's like a cheetah and a rabbit. The cheetah is the immune system. It catches the rabbit, it eats it. It's all over. But if the rabbit starts getting bigger over time, or develops spikes or something like that, now the cheetah can't catch it and can't do what it's doing. That's kind of what happens with the cancer and the immune system. The immune system is not changing, and the cancer is constantly developing new strategies. And the one that gets away, if that rabbit with spikes or something, it can just grow. It can outgrow it because there's nothing catching it anymore. So it just keeps growing, takes over the whole environment, and then that's kind of what happens with cancer. It gets away from the immune system, gets some trick, and then learns to do its thing. So it's constantly learning new tricks. And as we give these therapies, the same thing happens. If we treat with chemotherapy, maybe there's one that just has a mechanism that is resistant. So we kill all the cells that were susceptible, but the one cell that remained that had some mutation that allowed it to survive, it remains, and then it keeps dividing. Another new tumor that grows is resistant to that therapy. So it's the continued problem in cancer therapy, this evolutionary battle where the cancer is trying to learn new tricks with whatever selective pressure we apply to it, whether it's drugs, the immune system, or anything. That is the fight against cancer. We work out how the cancer finds a way to do it. We modify the drug to get around that thing. Cancer finds a new way, we find a way. And we hope that we, with enough options, there's two ways to win. We kind of back it into a corner where it has no options left and the drug just kills it. Or you kill it so quickly, or with two drugs, that all the cancer cells are killed and there's just no chance for the evolution to happen. It's when you have this long battle that there's this sort of chance for it to happen. So if you give two drugs, one of the cells needs to be resistant to both of them. And that's very, very unlikely. So there's a much better chance. It's the same with antibiotics. If you give two antibiotics, almost always works. If you give one, there's a chance that there's someone who was resistant to it.
For the antibody treatments, or just in general immunotherapy treatments, do men and women respond differently to them?
Great question. There was some papers very recently that testosterone is quite bad for the immune response to cancer. And I don't think in large trials it's played out that men and women have different responses to the immunotherapy. Men typically have a worse response to a lot of cancers. But there was some very recent papers that say that potentially testosterone is suppressing an important part of the immune system, and that has an effect on if you do well responding to immunotherapy, if you survive better long-term, and things like that. Maybe the testosterone effect on the immune system is kind of causing some of those effects we see in men having worse outcomes to cancer.
And do you know if it's true for viruses, like you mentioned influenza, but
yeah.
SARS?
Yeah, well, yeah, so there was some data that men were doing worse with the coronavirus. Previous to that, there's been quite a lot of data on influenza vaccines, and males typically have a worse immune response to the influenza vaccine than females, and the thinking is that they're less protected from the vaccine than women. And again, testosterone seems to be some correlate of if you have very high testosterone, sometimes your immune response is trimmed down a little bit, and then you maybe don't do as well with some of these things that rely on your immune system.
And well, for many years, most of the treatments were developed using men as the only volunteers. Yep. So is it different now?
Yeah, absolutely. Yeah, I think one of the biggest pushes of any clinical trial is to get equal representation of men and women, and then racial diversity as well. There's big genetic differences between races, different responses to different therapies. So in our trials, it's very important to sort of cover all those differences. And I think, I've only sort of maybe been involved with things like this for 10 years or so, but at least since I've been involved, that's been one of the top considerations as trials are designed. Will we get enrollment from enough different groups to sort of accurately cover the patient population that will eventually be treated? Prior to my time, it was absolutely what you said. Most of the genetic sequencing of cancer is, white male cancer. Many of the clinical trials are on things like that. So in my experience, at least, it's been better from what I know, sort of historically, it has been like.
We talked about the gender, how that influences the immune response, but does metabolic changes, like the cases of obesity or extreme skinniness on that side?
Yeah, exactly. There's a Goldilocks zone of weight for many cancers where, it was a great study. I think it was a UK study where they took the survival of almost every patient and correlated it with their body mass index. And depending on what your cancer was, you had almost like an optimal BMI. And it's it's more towards the heavier side of healthy. As you get towards the very underweight, you have an increased risk of dying from any cancer that you might get for almost every cancer. But the high side is also just as bad. As you start hitting these higher levels of BMI, there's a worse outcome for almost every cancer. And, it's a disease that needs a lot of energy. The cells are dividing very quickly, and if you provide them with what they need, they're going to divide more. There's many factors. Obesity has many things that it affects your body. It affects the immune system. So, very complicated there. But, almost every doctor says exercise more and lose some weight. So if we follow what the doctor says, it's probably a good idea.
Do you think it's ever going to be possible to prevent cancer?
Yeah, I hope so. I hope that we can detect it extremely well. There's amazing new data now on taking blood from patients and knowing that they have very small tumours in their body somewhere. So they can detect it far before we'd even see it on a scan or anything like that. So I think we'll get to the point where we can detect it very easily with very easy tests. Take a little bit of blood, know that a person has something. And then the second part of that is having treatments that we can use to treat it quickly. Every cancer is better if you treat it early. It's much, much better. The survival of patients with low-grade kidney cancer is well over 90%. And that's fantastic. And we want every cancer to be like that. We can catch it early. We know what to do with it. We have the tools. That's what the research is about, developing tools to let us, treat it in a way that maybe we didn't know how to do it before. Because as we find it earlier, we need new tools to kind of deal with that. So I think that's how we'll do it. I think we'll get on top of it. I kind of hope in our lifetime. I think, the last 10 years has been amazing. Every cancer is going down. The death rates, some of them are just very curable. You will be cured from your cancer. And I think if we can carry on with that, we'll be in a good place. There's big genetic risks to cancer. So for almost all the cancers, the first thing a doctor asks is, do you have a family history of cancer? And, breast cancer is probably the most important, BRCA1. This is a gene that when your DNA breaks, this gene helps repair it. And if you have particular mutations in your family line, your BRCA1 is a little bit less good at repairing the DNA than some of the other versions of that gene. And so over your life, you've got a little bit higher chance of getting some cancers because your particular version of a gene is a little bit different to other people's. It's not as good, and there's a higher risk of cancer. Maybe over time it's been a useful thing, because some of these mutations are how we evolve. Like maybe for some people it's a little bit better that they have a higher mutation rate, they're more adaptable to their environment. So maybe there was a good reason to have this, but now we definitely don't want that. So we can test patients' genomes, we can understand if they have high risk factors. But if you have a family history, it's very important to go to the doctor, starting around 40, get checked. The best treatment for cancer is catching it early. If you catch almost all the cancers early, they're very, very treatable diseases. And if you have a family history, you need to be a bit more careful and get checked often, because you probably have some genes that are not optimal at preventing cancer.
And for your own lab, do you have a lot of international students?
Yes. Yep. My lab is almost entirely international students and people. We have one student who's Ukrainian. We have a Polish student. Our lab manager is from the Czech Republic. One student from Venezuela. A scientist in our lab is from Vietnam. We have one American student. But people from all over. Really, science is very open to the world. People who want to do it, there's smart people all over the world, and they want to come and learn how to do things. So we're very open to that, and lots of international people in our lab.
And do you see a lot of cultural differences when people are working together? And is there any funny story you can tell maybe about it?
Good question. Funny story. We do have a lot of Eastern European students in our lab, and I don't know if the attitudes are correct, but they think that I'm very Australian, very positive. I think everything's fine and good. Our Eastern European wing have a much more they would call it a realistic view of the world, where they're very, I would say that things need to reach a very high standard before they're happy with how things are. And I think that goes really well with science. I think they bring a good perspective. I'm a very positive person, like to give a lot of positive feedback. They don't want the positive feedback until they've reached the sort of highest standards on the quality of our science. So there's a lot of jokes in our lab about that sort of stuff. But everyone gets on really well. I think it's a fun environment.
Yeah, that's great. I can totally relate to this approach. It's like nothing is good unless it's perfect.
Exactly. That's the vibe of our lab a little bit.
What fascinates you the most about your research? What makes you go to work every day?
The research is fantastic. Understanding cancer, how the genetics interacts with the immune system is very interesting. We look at massive data sets and terabytes and terabytes of data. Honestly, the thing that I like the most is the people. Science, it's really a group activity. There's no single scientist who is, some genius who cures a disease on their own. It takes discussion and talking and thinking about it from lots and lots of different people. You work with such incredibly smart people who have, these ideas that no one's ever thought of before. They see data from, a cancer patient or something, and they think something new that literally nobody has ever thought of before. And maybe it's a good idea, and maybe it's a terrible idea, but it's entirely new. And then we go and test if that is a thing that maybe ends up curing cancer. And just seeing that sort of level of, I think creativity is how I sort of see it, is just very inspiring to be around, particularly in young students. Sometimes they come in and they think their question is really stupid. They'll say something like, this might be a stupid question, but, and then you realize you can't actually answer what they're asking, and that what they're asking has really never been answered. And it's a really good question. And we need to go and think about it. So for me, it's the thinking and the people. Seeing students who have these ideas and then turn out to be correct is just very inspiring to be around, and I feel lucky to be around people like that. So that's what I come and do it for.
Great. Thank you for your time.
Cool.
Thanks.
Thank you. Cool.